Hi guys, I'm hoping that this is working. I'm trialing something new today, so please do bear with me if it doesn't work.
My feet!
Oh, no, I won't stop that. If it doesn't work, then I will switch over to the next part. My plan that I have. Okay. So I'm hoping that this is working. Guys, if you're watching on YouTube, can you let me know that you can hear me okay? And then I will get started. So if you can hear me, then that is great. And I will start my session. Hi guys, thank you. Thank you for joining me. Thank you. And okay, I'm just going to start going through some content for paper 2, obviously. Okay, so I'm going to display on my YouTube channel. So if you are joining me on here, then I will try my best to look at all the information that we have got. 1 second. I just need to sign in. Hold on. Sorry guys, give me 1 second. Just while I This is what happens when you switch from using a PC to using a Mac. Things change. There we go. This should hopefully make it work. There we go. 1 second. There we go. Right, I'm hoping that this is going to now connect. Oh, perfect. Thank you. You're telling me you can hear me. Right, I'm going to pop this out because it's not working the way I was hoping that it would work. And I will get started with actually doing the teaching side of things. Okay, so thank you for waiting, guys. I do appreciate that. You can go away and I will need to go back to... There we go. Right, I am back and I am ready to go. Okay, so in today's session, I want to talk about the genetics part of the course. I want to look at what's happening with the genetics part with inheritance, patterns of inheritance, how that can have a effect and what that therefore means for you if they're going to ask a question. So obviously we have population of a species and that is going to show variation. Now it's important that we understand what we mean by variation, and we understand that it is obviously going to be, in this case, if we're within a species, intra-specific variation. So I know last year's paper had a big question on that. Now that doesn't mean it's not that they finished with it, but it's important that we do note these key differences. So if we have a population of a species, we know that they are going to be able to produce fertile offspring. So we've got fertile offspring that are going to be able to be produced if we are going to have these organisms mating. So they're going to show variation. And what that variation means is these are the differences in observable characteristics. So when we talk about the phenotype, what you have to understand is that phenotype, what we can see, these observable characteristics, they are going to be what we use when we are looking at offspring, and then we can work back to what's known as their genotype. So the phenotype that we see is actually determined based on two things. It's based on the genetic material that they have, their genotype, but it's also to do with the environment that they live in. And so these two factors are going to cause variation. So whenever you're asked about genetics, you need to understand, well, what is an allele? So we talked in our last sessions about DNA. We've talked about the fact that the DNA is obviously going to be a sequence of bases. And if we're looking at a gene is a small section of the DNA molecule, and that is going to code for a protein to be produced. So when we do transcription translation, we are going to be transcribing the gene. So an allele is an alternate form of a gene. And so when we talk about alternate form of the gene, we have to remind ourselves that what we're looking at is actually the... One second, sorry guys. The allele being this alternate form means we've got a different DNA sequence. So if we have a different DNA sequence, that's therefore going to give us an understanding. As I said in yesterday's session, if you haven't watched it, I was talking about mutations, things like that. So if we have any form of mutation, a spontaneous mutation that's happened during DNA replication, that spontaneous mutation could result in a new allele being formed. So if we have any form of base change, if we have an insertion, if we have a deletion, then that is going to help us to understand the fact that there will be a difference in the bases that are going on. Sorry guys, give me a second. It's really hard when you're using something that you're not used to using. And so obviously I want it to be that you get the most. And if you have any questions or if you are needing any support, please do ask away, because that way I can see the chat, I can answer any questions that you have. Okay, so we've got this alternate form, we've got the fact that we've got a seed color, we've got the fact that we've got a petal color. So I'm going to look at these two just now. But the most important thing for you to note whenever we talk about patterns of inheritance is about whether there is a dominant allele and whether there is a recessive allele. So here I have a capital G. Now that capital G I'm using for green, and the capital P I'm using for pink. So they're either going to tell you which of the alleles is dominant, or they're going to give you the hint that the allele is dominant based on whether or not you have got a capital letter or a lowercase letter. So if I have a dominant allele, I always have a capital letter, and that tells me that that dominant allele is always expressed. And what we mean by it being expressed is that that is going to actively influence the organism's characteristics. Now what that means is that is going to be seen in the phenotype. So we need to understand that these proteins that we are making, they are involved in giving us our observable characteristics, whether that be the color, for example, in this case of the seeds, or whether that be the functioning of an enzyme, whatever that gene is. If we're talking about a dominant allele, we know that that is always expressed, that is actively influencing that characteristic if present. Now, a recessive allele, we always use the corresponding lowercase small letter. So if I have capital G for green, and I know that green is dominant over yellow, whenever I am showing the recessive trait, I know that I need to give the corresponding small g, but I must have two of them. So a recessive allele is only expressed if there are two alleles present. So in this case, if I have a heterozygous individual, because they have different alleles, they're heterozygous. Heterozygous means different. That will therefore mean that because I have a dominant allele, that's the one that's going to be expressed. That's the one that you're going to see. If I have a heterozygous for the petal color down here, then that means that I would have the capital P and the lowercase p, and that means the petal color would be pink if we have complete dominance. So what I'm going to give you is a little bit of an example that I find helps me to understand what's going on. So if I have, let's say, capital G, that is going to produce a functional enzyme. Okay, so if I have capital G, that is going to produce a functional enzyme. And if I have a lowercase g, that is going to produce a non-functional enzyme. Now, let's say I need to have a minimum of 25 functional enzymes. So I need a minimum of 25 functional enzymes for normal function to occur within the seed. Now, when I say normal function, what I mean is I have this functional enzyme. That enzyme makes sure that the chlorophyll is remaining, it's not broken down, and therefore my seeds are green. Okay, so if I have that, then the next thing I need to do is think, okay, if I've got capital G, let's say that causes the cells to produce 100 functional enzymes. So each of the capital G's causes 50 functional enzymes to be produced. Now, a cell will produce the enzymes, let's say, so therefore we've got 100. Now we only needed 25. Now we've got 100 functional enzymes, we definitely have green. So our seed color is green because I have 100 functional enzymes, but I only need 25, and that will ensure that I have a green seed. Now, if I have capital G, lowercase g, because I know that the capital G produces a functional enzyme, but the lowercase g, because of a mutation, we have variation, therefore we have these alleles, these different forms of the gene. Because I have these different forms, they have different primary structures. Those different primary structures therefore have different tertiary structures. Different tertiary structures means that there is the functional enzyme that has the active site that's complementary, or there's the non-functional enzyme which maybe has an active site that's not complementary. So in this case, I'm saying, okay, if I've got a capital G, I get 50 functional enzymes, and if I get a lowercase g, I get 50 of the non-functional enzymes. Through transcription and translation, we know that these proteins are going to be produced, these enzymes are going to be produced. I've got the functional ones, but I also have the non-functional ones. Now remember, I only actually required 25, no, let's try that again. I only required 25 functional enzymes. And so because I only needed 25 functional enzymes, I still get a green seed. It's not a darker green seed. It's that I just get a green seed because I have enough functional enzymes to keep the chlorophyll as it is within the seed. Now, if I have lowercase g, so if I have the recessive alleles and I have two of the recessive alleles, we can see in my fictional example that I would have 100 non-functional enzymes. So because I've got the two lowercase g's, I therefore have transcription, translation that's going to produce the enzyme. That enzyme is a non-functioning enzyme, and therefore it's not going to ensure that the chlorophyll remains. And there's nothing to mask it because there's no capital G. And so we see the recessive trait when we have two recessive alleles together. So the only time we would see a recessive trait in the phenotype is if the person possesses 2 of those recessive alleles. Okay. So I'm hoping that that is making sense to those of you who are still watching. And if not, please do ask questions as we go. It should be allowing you because I adapted it this time. Okay, so now what we're going to do is we're going to look at how we can inherit the alleles and how that can have effect on predicting phenotypic ratios. So we know Gregor Mendel studied the inheritance of these different characteristics of pea plants. We know that he looked at the pea plants. We know that he cross-pollinated them. We know that he looked at the phenotypes. He didn't actually know about the information beforehand. So he didn't have any of the information about chromosomes, about the DNA sequences. He was looking at the phenotypes and seeing what he could observe, and he noted it down. So if we were to take a mono-hybrid cross, and again, remember your keywords, mono means one. We're going to produce a hybrid, and if I have a phenotype which is a tall plant, and I take a short plant, and I therefore cross them, I would have all of my plants, all the offspring would be, if I can use the right color, 1 second, all of the offspring would be tall. So by looking at this, if I cross a tall and a short plant together, and if they are homozygous dominant, capital T, lowercase t, then all of my offspring are going to be tall. All of them are going to be heterozygous because what have they inherited? They've inherited their alleles from the parent plant. So if we look at this then and we say, well, that one is there and this one is there, we know that if we get 100% F1 generation where they are all showing the dominant phenotype, 100%, that would tell us that we have a homozygous dominant. Homo means same. They have the same alleles. So if I'm homozygous dominant, I have 2 capital T's. If I'm homozygous recessive, I have 2 lowercase t's. So I have the recessive alleles, or I have the dominant alleles, and I have 2 of the same because it's homozygous. So if we have this F1 generation, all of them are going to be capital T, lowercase t. And obviously if we then take 2 of the F1 and we cross-pollinate them, we would therefore do a Punnett grid, and that would give us our ability to predict the ratio. So you're coming down, you're coming across. So my genotype would be capital T. My phenotype would be tall. And we would do that for each of them, coming across, coming down. So each time we are coming down and we are coming across, and therefore we are getting our predictions as to what we would actually have. So obviously with a monohybrid cross, when I have 2 heterozygous individuals, I'm going to move this out of the way a second. So if I have 2 heteros, hetero versus a hetero, because I have my F2 generation, so I have taken 2 of these and put them in there, I would therefore end up with a 3:1 ratio of the dominant to the recessive trait. So now if we look at what's known as a test cross, whenever we do a test cross, what we're saying is a test cross is a way in which we want to see, is my phenotype, so if I've got a phenotype which is showing the dominant trait, so if I have a phenotype which is a tall individual, so if my plant is tall, so there's my tall, is it homozygous dominant for the tall trait, or is it heterozygous? So if we're trying to find out whether it's homozygous dominant or heterozygous, if we cross it with a heterozygous, no, let's try that again. Sorry, give me a second. If we cross it with a homozygous recessive, which is what we're going to do here, we're going to use a short plant. What we would then find is when we do the cross, what we're actually going to get with regards to our phenotypic ratio is slightly different. Let me just do that again. I had to move something. There we go. So what we're getting now is we are getting a 1-to-1 of being short. Sorry guys, you have to bear with me. I'm learning how to use my iPad rather than my computer, because I've got a Mac now and it's very different. Okay, so what we can see is, there we go. No, there we go. So we've got our tall here. So we have a 50% chance of getting tall and a 50% chance of getting short. So therefore we have a phenotypic ratio which is 1-to-1. So if we then looked at a dihybrid cross, so that's a monohybrid cross when we have one of the genes that we're looking at. When we're looking at a dihybrid cross, now what we're doing is we're actually looking at there being two. So di we know is about 2. So we're going to look at 2 genes. And at the moment, I'm going to assume that these genes are not linked. And I'll talk about linked genes a bit later on. So if I have the phenotype, and the phenotype here, I can see is that we have got yellow and round, yellow and round. So I've crossed 2 yellow and rounds, and I can see that I've got a capital Y, a capital R in my genotype, capital Y, capital R in my genotype. And so therefore I can say that yellow is the dominant and round is the dominant. And the reason I can say that is because when I have a heterozygous individual, and that heterozygous individual is showing the dominant trait, that means I can assume, so if they give it to you in the question, I can make an assumption that yellow is dominant and round is dominant because I have been told in the genotype that they are heterozygous. Okay, so that's one of the things for you to note. Now, through the process of meiosis, okay, and we'll talk about meiosis in a bit, but I'm going to remind you, when we look at meiosis, we have PMAT1, PMAT2. So through the process of meiosis, we are going to do a reduction division. So we're going to go from where there are 2 of each of the letters, 2 Y's, 2 R's, to having a haploid individual, in this case a haploid, as an individual, a haploid gamete, a haploid sex cell, and that haploid sex cell is only going to have one Y and one R. 1 Y and 1 R. So in the genotype of a diploid cell, we have got diploid. If it's a diploid cell, we know that it has 2 of every chromosomes. So 2 of every chromosome. So we had 2 Y's, 2 R's. Through the process of meiosis, we have done a reduction division. That reduction division now means that we have 1 of every chromosome. In this case, because we're assuming that they're not linked, therefore, if we're looking at this, I would say that the Y is on, let's say, chromosome number 1, and the R is on chromosome, let's say, number 4, just to make it up. So at the moment, we're saying that they are not linked. And because they're not linked, it means that we would get every possible combination. So when I am looking at only these 2 These are the two traits I'm looking at. These are the alleles and the genes that I want to focus on. I don't care about any of the other genes. I'm only focusing on these two. I'm focusing on the Y and I'm focusing on the R. And if I have a diploid cell, I would therefore have a homologous pair, and therefore what I would have is a chromosome 1. Because I've got homologous chromosomes, I have 2 chromosomes that are the same length with the same genes at the same loci. At chromosome 4, I have 2 chromosomes that are the same length with the same genes at the same loci. So what we're looking at is the color. The gene that determines the color is there. The gene that determines the shape is there. Now, I'm just making these up just to be clear. I'm not giving you actual real examples. I'm just trying to help it make sense. So you follow whatever they have given you in the exam question. So what we're going to do is we've crossed 2 heterozygous individuals. Through meiosis, we can predict what is the possible combination in the gametes. We know that they're haploid. We know they have to have 1 capital and 1 small, or 1 capital or 2 capitals or 2 smalls, depending on what they get. So there are 4 possible combinations. So 4 and 4. What we're then going to do is we're going to bring it down into my grid. Da, 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 da. And what you can see is the genotype and then the phenotype. And because we've got the genotype and then the phenotype, what I can then do is I can then use that to give my phenotypic ratio. So my phenotypic ratio is that we're going to have, in this case, 9 which are yellow. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9 which are yellow and round. I'm then going to have yellow and wrinkled, and I've got 1, 2, 3 which are yellow and wrinkled. And then I'm going to have, oh, 1 second. Then I'm going to have my, there we go, green and round. So there's my 1, 2, 3, and then I'm going to have my green and wrinkled. And so what we can see then is by doing a Punnett grid, by having a heterozygous cross with a heterozygous in a dihybrid cross, instead of a 3-to-1 ratio, I now have a 9-to-3 to 3-to-1 ratio. So that is the predicted ratio that we would expect, which is the phenotypic ratio. Now, if I were to do a cross and I was going to do another test cross, remember what we said before? The test cross would be that we're going to use a recessive. So in this case, the recessive trait is green and wrinkled. Now, in the exam, they will have told you what the recessive trait would be. They won't expect you to not know it. So this is why in mine, I know it's green and wrinkled. So if I've got green and wrinkled is my recessive trait and I do a test cross, I therefore know that during the process of meiosis, that reduction division, I'm going to end up with my haploid gametes, and those haploid gametes can only have one of each of the letters, 1 Y, 1 R. And so therefore, if it's got 1 Y, 1 R, they are then going to go into my Punnett grid. And this case, as we can see here, instead of getting my 9-to-3 to 3-to-1 ratio, I actually end up with a 1-to-1 I'll go pink to 1 ratio. So if it is a test cross and I have a heterozygous individual, my predicted phenotypic ratio is a 1-to-1-to-1-to-1 ratio. Now, they like to confuse you slightly in the exam, and they like to confuse you by throwing in, okay, we've got a yellow and wrinkled individual and we're going to cross it with a green and round individual. So let me, hold on, this is going to annoyingly switch everything around. I'll bring it back in a sec. There we go. So I've got a yellow and wrinkled, I've got a green and round. So because I've got a yellow and wrinkled and green and round, what I need to understand is that even still through the process of meiosis, I will get one of each. So I could either, in this case, get a capital Y lowercase r, or I could get a lowercase y lowercase r. When we look at this one, I can either get a lowercase y capital R, or I can get a lowercase y lowercase r. And so by doing this, I would end up, if I put that into a Punnett grid, still getting a 1:1:1:1 ratio. So if they're going to ask you to do a dihybrid cross, then do make sure that you know how to figure out whether or not they are going to be passed on, and also how we can then use that in predicting for the next generation. So if we look at epistasis then, epistasis we know is interactions of genes at different loci and how these are affecting the phenotype. Now here I have in my example my mice, my little mice that I drew in my Adobe. So if I have 3 different mice, so mice can either be white, albino, they can be black, or they can be brown in my diagram here. So straight away, I haven't given you any information apart from the genotypes and how they relate to the phenotype. So the first thing we need to understand is, okay, let's have a look at what we've got. We can quite clearly see in my information that if there is a lowercase c, if we have the recessive alleles, and we have 2 of them, because remember, if I go here, sorry, give me a sec, there, and if I go there, if I go recessive, I need 2 of them to actually show the recessive trait. Now, the recessive trait in this case is that it is going to have an effect. So C is epistatic to B. C is affecting whether or not B is being expressed. So we can see we have recessive epistasis here, because if the individual has 2 lowercase c's, that is having an effect on the expression of B. If we look at our black and our brown, then what we can see is if you have the recessive alleles for gene B, so recessive gene B, therefore will make you brown. So if you have 2 recessive alleles, you will therefore have the phenotype, which is brown. Whereas if we have 1 dominant allele or 2 dominant alleles, we will get the dominant trait. So therefore, in this example, I can conclude that black is dominant to brown because I can see the capital letters in my genotype, but also I can see that when they are a heterozygous, they are, if they're heterozygous for the alleles, they are going to display the dominant phenotype. So another question that they could ask is where they've given you an example and you've got to figure out. It really depends which way they want to go. Now, if we're looking at epistasis and if we're looking at the interactions of genes at different loci, we've got things like a repressor protein. We talked about the repressor protein in my session yesterday, in the fact that if I have a repressor protein that is going to bind, and that repressor protein binds and blocks the RNA polymerase from binding. That will therefore prevent transcription occurring. So that's one of the things that could happen. It could be that the gene is going to code for an inhibitory molecule. Now, that inhibitor molecule could bind to an enzyme involved in a pigment production pathway, or it could be that it's the production of a non-functional enzyme. So if we have a non-functional enzyme needed for an intermediate molecule, then if we stop that from being able to be converted, we stop the pathway. So it's really important that you read the information that they've given you in the question to figure out what it is that they are trying to give you the hint of. And I'm going to actually do a question now to show you this. So one of the things that they could say is, here we go, the color of pea plants, and we've been told about their flowers, is controlled by two genes. It's controlled by gene C and it's controlled by gene P. So not only have we been told about the color, but they could actually talk to us about classification. There's my genus, there's my species. How do I know? I've got a capital letter in the first part of the name, and it's a lowercase letter in the second part. We always, for the international naming nomenclature, we do this with genus and then species. So the color of this pea plant, this specific species of pea plant, is controlled by two genes. So now we're being told that if we've got the dominant allele, which is P, and it's going to code for the production of a purple pigment. So if the dominant allele P is present, we would expect purple pigment. Now, if it was a monohybrid cross, we would obviously know that we would be looking at it being capital P, or capital P, lowercase p, and we would get a purple flower. And if we don't get a purple flower, then what do we get? Well, they've told us if we have the homozygous recessive alleles, we are not going to produce the pigment, we're going to get white flowers. So therefore, we know that because we're going to get white flowers, that is going to therefore be my lowercase p. So I always, even though they've told me that information in the stem of the question, I always try and write a little note to myself, because if I've written it down, I've then processed what I've read. Sometimes they can give you lots of words and it can get a little bit confusing. So we're then told later on that the dominant allele C is required to activate the pigment production pathway. The recessive allele C means there's no pigment regardless of whether we have got capital P present or not. Now, what they could decide to do in the question is they could actually say, where's the evidence from the information that we've given you to say that the dominant allele for the color purple is actually caused by the capital P allele and not the recessive allele. So how do you prove that purple is dominant over white? You prove that by it saying, when we crossed a white flower pea plant and a purple flowered pea plant, what we got was that all of the offspring were heterozygous and they had purple flowers. So when we crossed a white flower pea plant and we crossed a purple flowered pea plant and all of the offspring were purple and they were heterozygous, that would therefore indicate that the parent plants were both homozygous. Now, I've given a really generalized conclusion there, because all 16 of the offspring, actually 16 is quite a small number. So 16 offspring drawing a conclusion that says that they were both homozygous, and homozygous dominant, homozygous recessive, because all of the offspring were heterozygous. That is generalized because there's only 16. If I've got thousands and all of those thousands of offspring have the purple phenotype, then yes, I can draw that conclusion. So do make sure when you're looking at this that you are considering the information, because it could be an evaluate question about the scientist concluding. Okay, so we're going to move on, and we've got 2 heterozygous purple pea plants that are going to be crossed, and we're going to use a genetic diagram to work out that phenotypic ratio. So we know we've got purples. We know that we have got heterozygous individuals for the purple pea plant, and therefore we can work out our gametes, and we can then draw a Punnett grid. So what are we actually talking about? What's actually happening? Well, if we look at it here, capital C, and it doesn't matter what I have here. So I've drawn it with a little line underneath because it could be the fact that we have got capital C, or it could be that it's capital C, lowercase c. It does not matter. So that's why I've drawn it as a little dash underneath. And if I have capital P again, it doesn't matter if I've got capital P, or if I have got capital P, lowercase p. If I have capital C, at least one capital C and one capital P, I will end up with purple flowers. So what I'm saying is my colorless molecule, let's say genotype C codes for the production of enzyme number 1, a functional enzyme. With this functional enzyme being produced, what's going to happen is that is going to change my colorless molecule into an intermediate molecule. What we're then going to have is my intermediate molecule is then going to be converted by enzyme 2 being present, another functional protein, and that will therefore result in that purple pigmentation being produced. Now, if I have capital C, again, doesn't matter if I've got capital C, or capital C lowercase c, if I have 2 of my recessive P alleles, I know that that means that I'm going to produce enzyme 1. Enzyme 1 is functional, so my colorless molecule will be able to be converted into an intermediate molecule. However, because I have got my 2 lowercase p's, and I was told that the recessive, homozygous recessive, do not produce the pigment. So because they don't produce the pigment, I cannot actually convert it, and therefore my flowers are white. So if we look at having the recessive alleles for the C, and this time doesn't matter whether I have got capital P, or whether I've got capital P, lowercase p, I'm still going to end up with white flowers. And the reason being is I'm not this time producing enzyme 1. So because I'm not producing a functional protein, a functional enzyme, the tertiary structure isn't complementary to convert the colorless molecule into a intermediate molecule. That therefore means that even if I am able to transcribe and translate and produce a functional enzyme 2, I still don't get a purple pigment because I don't have the pathway to produce the intermediate molecule. So it really just depends which way they want to go in the exam question. So if we understand pigmentation, whenever we're talking about pigmentation, involves both structural and regulatory genes. And those structural genes are normally the ones that make the pigments, while the regulatory genes are the ones that control where and when it's going to happen. So if we think about zebras, for example, how is it that zebras have the stripes And obviously this striped pattern makes them able to confuse their predator. That helps them to survive, and that survival means that they can reproduce, and that reproduction can result in passing on the alleles, which are advantageous alleles. So if we understand that our genes can be ones that are going to be producing the pigments, but also we have others which are regulatory genes that will control where and when the different pigments are made. That therefore means that we can hopefully understand epistasis. So going back to our original example, if we were to fill in our Punnett diagram, what we would then get is we wouldn't get our expected ratio. When we've got heterozygous to heterozygous, we expect there to be a 9 to 3 to 3 to 1 ratio. But that's not what we've got, because what we've got because of epistasis is we have actually got 1, 2, 3, 4, 5, 6, 7, 8, 9, which are giving us the purple flower. And then we have 1, 2, 3, 4, 5, 6, 7, which are giving us the white flower. So we get a different ratio, a different predicted ratio based on the information that we have just looked at, being able to go through the process of what would we expect, what would we actually get if those genes were not linked, and if we were to cross two heterozygous individuals, and therefore if there is any type, in this case of epistasis, the interactions of one gene on the expression of another gene. So the expression of another gene doesn't mean that it is necessarily about the preventing it from producing the protein. It could be, because it could be a repressor protein, but it could be about stopping the pathway for the production of that phenotype. And the phenotype was the purple flowers. So the presence of the recessive allele C, where there's no pigment being produced, that is having an effect on our phenotypic ratio, because we're getting much more that are showing the recessive trait, when actually it's because they also have the recessive alleles for C, not just for P. Okay, so that's about epistasis. And if they're going to give you an example, it will be a one where the information is in front of you. Just got to slowly work your way through the example. So what do we know about inheritance, and what do we know about the variations that we get? So we know that we have got obviously meiosis, we know we've got mitosis, we know that we've got diploid cells, we know that we've got haploid cells. So we know through the process of meiosis, we are going to be able to take our diploid cells, so our 2N, our diploid cells, and through the process of meiosis, we are going to reduce them down to haploid daughter cells. Now, the key thing to note is whenever we are doing a reduction division, before we can do mitosis, before we can do meiosis, we need to have DNA replication occurring. So we need DNA replication to occur. We need there to be our sister chromatids, and we need those sister chromatids to be attached by the centromeres, because we need our spindle fibers to attach to the centromeres, and we need those spindle fibers to push and pull and move them during metaphase 1 into the equator. So we know that we have done DNA replication. We know that we have our sister chromatids. We know through prophase, metaphase, anaphase, telophase 1, we are going to produce 2 daughter cells, and those daughter cells are already going to be haploid. They are haploid, but they are made up of sister chromatids. I really need to practice writing on my iPad. Okay, so if we understand that, we should know through the process of meiosis that we are going to have prophase 1 happen, and during prophase 1 that will result in the potential crossing over occurring because our homologous chromosomes are going to come together as bivalent. So here we have my homologous chromosomes, my chromosomes which are the same length with the same genes at the same loci. They are going to be together and they are going to be moved in their pairs. Okay, so that's the first thing about prophase. Now we know that meiosis is about producing our sex cells because we know that we're going to have sexual reproduction occur. So sexual reproduction is where we're going to have this combination of genetic material from two parental sources. So in this case, we've got a mum and a dad, we've got an egg, we've got a sperm, each of them containing a nucleus, that nucleus that is haploid, and that haploid nucleus being produced through the process of meiosis. So through sexual reproduction, this combination of the genetic material in the production of those gametes, we are going to produce variation. And that first bit of variation could be happening because of crossing over during prophase 1. So crossing over during prophase 1 can only be talked about if we're looking at the joining of our genes. So let me just, I'm going to move this bit up a second. No, I want it to go there. Right, so we're going to talk about crossing over. We're going to talk about how that actually happens, but the crossing over only happens during prophase 1. Independent assortment, how do they line up? That happens during metaphase 1. So in my diagram here, I've got blue and then I've got a long red. I've got a short red and a long blue. Did they line up blue and blue on the one side and red, or did they line up, as you can see here, with an alternate combination? So how did they line up at the metaphase plate at metaphase 1? Genes from different traits are obviously going to be distributed differently depending on how that combination is lined up. During the process of anaphase 2, what we've got is anaphase 1, our homologous chromosomes were being separated, but during anaphase 2, our sister chromatids are being pulled apart. So because our sister chromatids are being pulled apart, that's going to result in a potential different distribution of the genes in the gametes. I think all the students are in the chemistry exam. Will this be available later? Yes. So to answer the question, yes, this is being recorded right now on my YouTube channel with the display of what I'm talking about as well, and it will be available for those. I know there's that's the reason I'm doing this, because my students that I tutor privately are all in their chemistry exam, and they'll be in their maths exam tomorrow. So I'm going to do today and tomorrow, and then I'll be seeing some of my students later on. But yes, so for anyone who has a student who is currently sitting their math, their chemistry, their business, or I think it's sociology is today, any of them sitting those exams, these are available for you to use later on. Okay, so back to this. Crossing over, independent assortment is a way in which we are going to potentially promote variation. And obviously we've got the random fertilization. So which of the male gametes fuses first with the female gamete, and which of those nuclei, those combination of alleles, are coming together. And so that's how the variation can actually be promoted. Now, last year, the question they did on variation was about intra and inter-specific variation when you were looking at your sales, whereas I feel like they're potentially going to come down the route of still looking at variation, but looking at what could cause it. And obviously you have got the mutations that can occur, but you have to remember there's so many other things as well. So make sure you understand that yes, you have the mutations that can cause different alleles to arise, and that is spontaneous mutations, but we also have to remember that variation is also caused through crossing over, independent assortment, random fertilization through this process of sexual reproduction. So if we're looking at sex determination when it comes to biology, we know we've got our sex chromosomes. And I know they did a question on this and they gave you a random example, but I think they've not finished yet because they're potentially going to look at a sex-linked gene in this year's exam. So I'm going to look at the linkage in a minute, but I'm also going to look at sex linkage. So they're either going to give you it as linkage on its own, or they're going to give it to you as a sex-linked gene. So it really just depends which way they decide to go. We know through meiosis we're going to end up with our gametes being haploid. So if it's female, you get one of each of the chromosomes, and in humans we obviously have 23 pairs. The 23rd pair of chromosomes are your sex chromosomes. So whether you get an X or an X, because obviously you can only get one haploid, you get an X. When it comes to the males, because they have different sex chromosomes, they have an X and a Y, that therefore means that we can determine that if you are biologically female in the fact that you have an X and an X chromosome, that therefore meant that you actually had the sperm cell that contained the X chromosome that fused with your mother's egg cell. So we can do some predictions based on that if we understand the fact that the combination that you get, whether you're XY male or whether you're XX female, had to come from the inheritance, the chromosomes that you inherited from your parents. So obviously if we have a female and a male, we therefore can do a prediction of a phenotypic ratio of 1:1. So they could ask you to do the phenotypic ratio of sex determination, or they could ask about how the sex determination would be with also adding in the inheritance of a trait. So if we are looking at, for example, let's say our red-green color blindness. Now let's say we've got red-green color blindness. So here's my little diagram. If I have red-green color blindness, then I should know that that is a sex-linked trait. It is linked to the X chromosome. The X chromosome being larger means it's got more genes on it. And therefore, if you are unaffected as a female, you would have two capital B's. We're going to use the capital B for blindness, color blindness. They'll give you an example of what they're wanting you to use as the letters. If I am a heterozygous, I am a carrier. And if I am if I am affected by it, I would have to have two recessive alleles, because remember, my dominant allele will mask the presence of a recessive allele. So if I am male, I would therefore have one X chromosome, therefore one capital B, I am unaffected. If I am male and I am affected, I only require one of the alleles, because there's no masking going on. There's no other allele to mask the presence of that one. Oops. Okay, so what does that mean then if they give you, and again, I do, which I could be wrong, I have a strong feeling that they're going to give you some form of a family chart, and they're expecting you to be able to pull out from this kind of information. So the first thing I would do, let me just take you slowly through because I was doing a bit of this with my private clients. And so what I would say, the first thing I would always do is I would start and say, well, quite easily I can determine whether they are X and Y. So the first thing I would do is start with my males, X, Y, X, Y, X, Y, X, Y. So that's the first thing that I'm going to do, is note where my males are. And then I'm going to note where my females are. Okay, X, 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 X. Now they will give you a key that tells you that the circles are female and that the squares are males. They'll give you a key that says that the shaded ones have the trait and the clear ones don't have the trait. So now we need to use that as our understanding. So we know, because they'll have told us in the question, that the affected color blindness is a recessive trait. So they're going to tell me somewhere that this is a recessive trait in the stem of the question. So therefore I know to have the disorder, I would need to have a little b. I would need to have a little b. I would need to have, with my X's, 2 little b's, 2 little b's, 2 little b's, 2 little b's. So I can say straight away, throw my pen. I can say straight away that the Y for that male came from the dad, and I can say that that B came from the mum. I can say that again, that B came from the mum and that Y came from the dad. Now I know that the dad does not have the trait, and so therefore, because I know the dad does not have the trait, there is a capital B. So I know for sure that that female has at least one capital B, because they inherited that from their dad. So because they inherited that from their dad, they at least do not have the trait, because their dad passed on the dominant allele. Now we know that the mum has got the trait, and therefore we know that the mum has passed on the lowercase b. So we know that, and it doesn't matter which one we're talking about, we know that they have inherited one of the lowercase b's. Now, what we can determine is that the females further down, what we've got is that yellow has come from at least the dad. But where did the other recessive allele come from? So the other recessive allele that they have got, this one here, that had to come from the mum. And so therefore we can determine that the mum is going to have at least one of the recessive alleles, because they've passed that on to the daughters. And the fact that they don't have it means that they have to have a capital B. So then we can look at, okay, they have to have a capital B, and that is what they passed on to their sons. Because remember, their sons were getting the Y from the dad. The Y from the dad. So therefore, the sons have inherited the dominant allele from the mum. So it's really important when you're going through a pedigree chart like this, that you understand that the square and the circle with the line going between, they are mating, and the offspring that they have produced are the ones that are on the lines coming down. And you should then be able to work out the genotypes of those individuals, and you should be able to use that to confirm whether there is a dominant trait or whether there is a recessive trait, whether it is sex linked or whether it is not. Now, if I were to change, let's say I decide to change this one, and I am going to, hold on one second, change that color, and I'm going to make that now that this one does not have the trait. If they did not have the trait, that completely changes what I would have here. So if they don't have the trait, that now means that they have to have a capital B. But I can determine that the fact that they have a offspring, the son that has the lowercase, the recessive allele, the lowercase b, I can say that they have to be heterozygous for it because of that lowercase b. So it really does depend what they are going to give you in the stem of the question, whether it is color blindness, whether it's hemophilia, whether it is sickle cell anemia. It really just depends the example that they give you. It's about reading the stem of the question and then using that to guide you in that thinking, because you should know that you are going to get a combination of the alleles from your parents, and therefore if there is a trait, whether it's dominant or whether it's recessive, that trait has to be passed on. And so you can work through the pedigree chart, you can look at the alleles that are being passed on and use that to guide you on whether or not the mum was heterozygous or homozygous for a trait. Right, I'm going to pause for a minute and I will come back. So if you just bear with me, I will be back in a second. Okay. Right, so let's move on. Oh, 1 second. I have no idea what's just happened there. Never mind. You can just have it as a random green screen. Okay, so let's move on with the next one then. So autosomal linkage. So they could ask a question about there being a... Oh, it's going to bug me. 1 second, guys. Let's just... Never mind. Autosomal linkage, they could ask you about sex-linked genes, or they could ask you about autosomal linkage. Now, if they're asking you about autosomal linkage, then what you need to note is that the autosomal linkage is them saying that it's on the same chromosome. So we know in humans that we have our chromosome 1 to chromosome 23. So we know we've got chromosome 1 to 23. And so therefore we cannot, okay, just to be clear, we have already eliminated out our sex chromosome. So if it is a sex-linked gene, then that would be the sex-linked gene is talking about it being linked to the X chromosome. They may even throw it in there saying it's linked to the Y chromosome. They do sometimes like to shake it up a little bit. If they do, then again, that would be application. It will be in front of you, the information that you need to be able to answer that question. Hi, just wanted to ask if all of your Module 6 videos are complete on your channel. Yes, all my videos are complete on my channel for Module 6. So yes, they are all there if you want to go and watch. They're shorter videos, and they are using the resources that I've got. Obviously, they are the ones that I've made over the last year, year and a half actually, preparing you for this exam. So yes, they are complete and they are all there. Okay. So going back, and thank you for writing here, because I do appreciate when people actually engage with what I'm doing. I can then answer the questions. Sometimes I feel like I'm just sitting here talking to myself. So I hope you guys appreciate it. Yeah, let's keep going. Right. So if we have a linked gene, we need to understand that these linked genes are inherited as one unit. So when they are going to be going through the process of meiosis, there would be no independent assortment if we have autosomally linked gene. Now, that means that unless during meiosis there is some form of crossing over, so if the genes are far enough away from each other on the chromosomes, then potentially there can be crossing over occur. So let's just have a look at what that means. So here I have my chromosomes, and we're using chromosome number 4. We're imagining that these are our chromosomes. On chromosome number 4, we've got our genes and we've got our heterozygous individual. So I've made them specifically heterozygous individuals because I want that to be in your head. Remember that they can be homozygous dominant, they can be homozygous recessive. If they're homozygous dominant, homozygous recessive, if crossing over occurred, it's not going to do anything. The only part that crossing over matters with is if you have a heterozygous individual. So if I have DNA replication occur, if I have the sister chromatids and they're held together at the centromere, what can then happen is during prophase 1, when they pair together as bivalents, what they're going to do is they are therefore going to potentially form chiasms. Now, a chiasm is a point where there will be an exchange. So if during meiosis 2, they are being separated, not if, they will be separated, during the anaphase 2 of meiosis 2, we know that the sister chromatids are going to be pulled apart. Well, actually, during anaphase 1, when the chromosomes were being pulled apart, they'd already exchanged the material. So if we are looking at the chromosomes, what you would get, if I quickly do this, hold on a second, if I could get rid of that. I've got to remember, I'm using a Mac now, and so it's very different with my shortcuts. My shortcuts don't work in the same way. What am I looking for? I'm looking for my Patterns of Inheritance study guide, because I have a really nice picture on there. So give me 1 second whilst I pull this through. Patterns of Inheritance study guide, where are you? Module 6. That's what I want. Right, give me 1 second. I just want to bring through this one. Okay. So, oh no, I did it again. I've got to remember it's Command. You have ever used a PC for the majority of the things that you do and then you switch to a Mac, you will know why my frustration is happening right now. Because it is one of those things that I'm so used to doing things really quickly and I'm having to relearn. But that's not a bad thing. Sometimes it's good. Right, okay. So I'll come back to this in a second. So if we are looking at the process of meiosis and we're looking at are bivalent. So in this example here, I've got my bivalent, I've got my homologous chromosomes, and if no crossing over were to occur, I'd end up with the same chromosomes. And so if we're looking at in my example, which was A and B, wasn't it? So my example above was A and B. So if I had my A and my capital B, got my A and my capital B, I've got my lowercase a, lowercase b, lowercase a, lowercase b. No crossing over is going to occur. During my prophase 1, if crossing over were to occur, I had my A, I had my B, I had my A, and now I've got my lowercase b. I have my lowercase a, my capital B, my lowercase a, my lowercase b. So what you should know is that crossing over doesn't always occur. The key thing about variation and what would add to variation is that if crossing over occurs, the crossing over has to occur during prophase, so prophase 1, and then during the metaphase 1, they're lining up at the equator. During anaphase 1, as they're being pulled to the opposite poles, they are therefore going to be separated. These homologous chromosomes, they'll have already exchanged the information. And so if we go back to this diagram here, if crossing over occurs, then during meiosis 2, when those sister chromatids are being pulled apart, what we've now got is we've now got recombinants. We have got variation because before we only had, in our example before, we had capital A, capital B. Now we've got capital A, lowercase b, lowercase a, capital B, lowercase a, lowercase b. So if crossing over has occurred, then we would end up with variation because we'd end up with recombinants. The recombinants are different from the parental chromosomes. So a recombinant offspring will have a different combination of alleles from their parent. And we can look at the recombination frequency by saying, right, well, the number of recombinant offspring divided by that total number of offspring. Okay, so if we are going to look at this, if we're going to look at inheritance, whether it is a monohybrid cross, whether it's dihybrid cross, whether we are trying to identify whether there is a significant difference between, in this case, if we're going to use a chi-squared test, we are looking at whether there is a difference between the results that we've observed and those that we expect. So when we did a heterozygous with a heterozygous, if it was monohybrid cross, we'd expect a 3-to-1 ratio. If we're doing a dihybrid cross, we'd expect a 9-to-3 to 3-to-1 ratio. If we were doing a test cross and we were looking at heterozygous versus a homozygous recessive, in a monohybrid cross, it would be a 1-to-1 ratio. If we're looking at it with a dihybrid cross, it'd be 1-to-1-to-1-to-1 ratio. So if we can do a prediction, if we have a way in predicting the ratio, in this case we do because we have our genetic diagrams that we can do and we can therefore predict those ratios, then what we would expect is the information, null hypothesis that's been given. There is no significant difference between what we expect and what we observe. Any difference we do see is due to chance. So we know that you'll be given the chi-squared test, the equation. We know that you would then be given some information. So in this case, I've given you 2 heterozygous purple flowers with long pollens were crossed. So again, using that information, we can say, well, they are heterozygous and they're purple and they're long. Therefore, purple and long is the dominant trait. So if purple and long is the dominant trait, then I know that my red and round must be my recessive traits. So therefore, I can use this information to figure out where my ratio should be. So my expected ratio is a 9 to 3 to 3 to 1. So the first thing I need to do is I need to add them all together. So if I've got 6,952 is my total, and I know my expected ratio is 9 that are purple and long, 1 which is red and round, and therefore these two should both be at 3. That would therefore indicate for me that I have got my number, which is my total, divided by 16, which is 434.5. Now, just be aware, if they decide to try and trip you up slightly, remember it's not just a 9 to 3 to 3 to 1 ratio. It could be that they are giving you a test cross. And if they give you a test cross, our expected ratio would be 1 to 1 to 1 to 1. So if that's our expected ratio, we would therefore take our 6,952 and we would divide that by 4 to give us what our 1 to 1 to 1 to 1 ratio was. So it's really important that you look at the stem of the question and that you are looking at what it is that they are asking and what they are telling you. Now, if they tell you that they are linked genes, we would actually treat them as if we were doing a monohybrid cross. But obviously, we know that because of crossing over, they can still act like a dihybrid cross. So we would still have our predicted ratio, and therefore we would use that to work out that obviously that's our divided by 16, times it by 9. So our expected purple and long was 3,910. Our expected for both these two was 1,309. And our obviously expected for our red and round was 434.5. So we have those numbers. Because we have those numbers, we can then put that into the equation and we can then work out what our chi-squared value is. So our chi-squared value is 3,371.5913. So once we've got our calculation sorted, once we've inputted the data, we would therefore be able to then use our probability table. So our null hypothesis, remember, is there's no significant difference between the expected and what we observed. We've calculated our chi-squared value. Because we have 4 categories and we know it's n-1 for our degrees of freedom, we look at 4 minus 1, which is 3. So our degrees of freedom is 3. We obviously go to our p-value of 0.05. So we always make sure if we're going to do this that we know where we're going to, and therefore our critical value is 7.82. So our chi-squared value is huge. It is significantly higher than the critical value of 7.82. Therefore, the difference between our observed data and our expected data is significant. Also, if we were to move down here, our value is so huge, it's even significant at that value of a p. One second. If we go down here, we're even greater than there. So as I've always said, draw a little star as to where your number would lie that you've calculated. So where the number would lie is obviously greater than that. So we are saying it's also significant at a p of 0.01. So therefore 99% certain that the results are not due to chance, that the results are due to the fact that these are linked genes. So we are therefore rejecting our null hypothesis, and we can see that because of the way that they have interacted, that they are most likely autosomal linked. Oh my word, it's doing it again now. So if we're thinking about the autosomal linkage, we therefore need to bring that into play. There we go. Right. when we're answering the question. Now, it could be due to epistasis. So that's the other thing that we need to consider. It could have been due to epistasis. It just depends what example they're giving you. In this case, we can see that these two are much lower. And if we are considering our dominant and recessive, they are different. These are recombinant offspring to the parental offspring. So hold on, let me just add in another one. So what I want to show you, just to be clear, is when we are looking at our traits, if we were doing, let's go our A and B. So here's my A and B, and then I'm going to use my A and B, my A and B. Now, if crossing over occurred in a homozygous dominant, doesn't matter. If crossing over occurs in a homozygous recessive, it doesn't matter, because what is the offspring potential going to be? Well, it's dependent on the gametes. So therefore the gametes would be that, or they would be that. Okay. I really am still learning how to use this pen on here. So there we have a gametes. The gametes are either going to be capital A, capital B, or the gametes are going to be lowercase a, lowercase b. If we were to cross those two individuals, we would therefore get a capital A, capital B, lowercase a, lowercase b. Now, if we actually draw this as being linked genes, then we would draw it like this, A and B. Now, if crossing over were to occur, in this case, we would potentially get a difference. So this is where the only time that crossing over will have an effect is if you have a heterozygous individual. Now, if crossing over were to occur, then that's when you get your potential of capital A, capital A, capital B, capital B, hold on, capital A, capital B, doing this all wrong now because I'm trying to do it a different way, capital A, lowercase b, then we've got lowercase a, capital B, lowercase a, lowercase b. Does that make sense? Yes, 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 yes. So there's our four possible A and B, yeah, gametes that we could get if crossing over were to occur. Because remember, if I start with my A and B, one thing that I haven't done is I haven't remembered that DNA replication occurs. And that DNA replication occurs resulting in my sister chromatids A and B. And so what we're actually saying is that if crossing over has occurred, if I rotate that slightly, it's occurred like that. And so when they're separated, that's why you would potentially get your A and B. You would potentially get your A with a small b. You would get your this or this. So that's my A and B. That's my A. That's my B. That's my B. That's my A. That's my A. That's my B. So if crossing over does occur, then that's what we're looking at here. But we have to be sure that we are noting the information they've given us in the stem of the question, because it really does just depend. Okay, so moving on then, variation we know can be discrete. It can be discontinuous. It appears in specific values. So your blood groups is a prime example. So I am blood group O. I am O negative, and when you guys are sitting your third paper, I'm going to go and give blood, and I'm going to give my 50th donation of blood. So excited to hit that 50 mark. because they need my blood and I've not been able to do it for a long time. So I am O negative. And it's a funny one because I say this all the time. My mum and dad, it used to be when you were born, they would take a sample of your blood and they would test your blood to see what blood group you are. And my mum is A, my dad is B, and yet I am O. So because my mum is A and my dad is B and I'm O, I know both my mum and my dad are carriers for the recessive allele, which is O. And I have inherited both of those recessive alleles because they are, my mum being A, so she is A, my dad is B, and therefore she has IA, IO. So it can either be written like that or they could do it as a little lowercase one. It really depends on the exam paper. My dad is IB and he is also O because I obviously had to inherit one from my mum and I had to inherit one from my dad. And so therefore they both have the recessive allele for O. They both pass that on to me and I am O negative. So because it is controlled by either just one or two genes, depending on what they are going to ask you a question of, whether it was female to male, whether it's pea shape, because they can do it with plants, whatever it is that they're telling you, if it is discrete, if you can be put into a category and that is it, there's no intermediates in between, you're either A, B, or O, that is where we show variation, but that variation is discontinuous. So we would represent that in a bar chart. Now, if we're looking at the alleles, so the percentage of the population with the alleles of homozygous dominant, heterozygous or homozygous recessive, what we can see is they follow a normal distribution curve. And we can see that there is selection for the heterozygous. So if we were to do a Hardy-Weinberg equation on this, we would see that the population would be a lot higher for being heterozygous. So our 2pq in a population would be greater than our p squared and our q squared. Oh my word. And so therefore we know our Hardy-Weinberg is p squared plus 2pq plus q squared, which is equal to 1. So we know our p plus q is equal to 1. Okay, so if we understand about discrete and we understand about it being homozygous dominant, heterozygous, homozygous recessive, and we can look at the population, percentage in the population, we can look at whether or not that is stabilizing. If it's stabilizing, then we can use Hardy-Weinberg's equation. If we are looking at continuous data rather than discontinuous data, let's have a look at the finches. Now, we know that finches have got beaks that allow them to eat the food that they are going to feed on. So if we're looking at the seed-eating birds of these finches and we're looking at their beaks and the ability for them to break open the seeds that are available, then we need to understand that the depth of the beak is actually under what we call polygenic control. So it's under polygenic control, it's under the control of 3 genes. Now, each of those genes, A, B, and C, let's call them, have got 2 alleles. Now, if you have the dominant allele, that's going to add depth. If you have the recessive allele, then no depth is going to be added. So what you can get is the combination. Remember, through meiosis, we get a haploid individual. When they fuse together, we get a diploid cell. So what we can see here is if you have all dominants, if you are homozygous dominant for all 3 of those genes, you would therefore have an added depth of 6. If you are homozygous recessive for those, you would have an added depth of 0. So what you can say is AB, capital, lowercase c, we would therefore have 1, 2, there's nothing there. And on the other side, 1, 2, nothing there. So you can see how this works. So if I've got 3 genes rather than a dihybrid cross, I've now got 3 genes. It's making it a little bit more complicated, but basically what are we showing? We're showing that those 3 genes, the A, the B, the C, with 2 alleles at each loci, if we were to graph them out, we would actually see a normal distribution coming through. So continuous variation, where we have this range of phenotypes, in this case for the finch is about the depth of their beak. Is the depth of the beak large or is the depth of the beak small? No, you do not need to know any case studies. You don't have to memorize any case studies. It's more what they're looking for you to do is be able to apply your knowledge to the question that they've decided to give you this year. So there will be all the information you need in the stem of the question. You just have to be able to find it, and you have to then apply your knowledge that you have about whether it's discrete, whether it's continuous variation, whether there is a range of phenotypes, or whether there is not in the case of discrete. So what we know is when we look at continuous variation, because we can have any values in between, what we see is a normal distribution. And when we see a normal distribution, if we're looking at heights of trees, the weight, your foot length, what we will see is that this is caused by multiple genes interacting, as well as the environmental factors. Whether you have blood group A or B, the environment has no effect on it. You don't have A or B based on your environment. You have A or B based on the genes that you possess. Are you able to roll your tongue or not? Do you have dangly or attached earlobes? Do you have a widow's peak? Do you have the double crown at the back? So I have 2 daughters, one who has a double crown and one who does not. So therefore the genes that they've inherited are different, and the interactions of those genes are going to be different in why we see variation within families. So my eldest daughter has more of my traits than she does of my partner. However, my youngest has more of my partner's traits than she does of my of me. However, when you look at them as they are growing and developing, they are obviously also being influenced by their environment, by the interactions that they have. So your genetics are the genotype, the alleles that you possess. The phenotype, the physical characteristic, is the combination of the genes along with the environment. And if they look at, for example, a question when I talk about your immune system, the genetics that you have come from what you have inherited, but then you have the exposure of the environment. Which pathogens have you been exposed to? How does that have an effect on your immune response? Which is what they could go on and look at. But coming back to this and coming back to your phenotypic variation, obviously your phenotypic variation can be caused by differences in the environment. So if we're looking at animals and the body mass, if we look at the climate, so obviously it is crazy right now. It keeps changing. Yesterday we had a thunder and lightning storm here, and then it was glorious sunshine. It's glorious sunshine today, and yet there were dark clouds. So it's all about how could your environment have an effect on, for example, an organism. If you have a hotter climate, if it's hotter, are you therefore, and this would be a paper 3 question, are you therefore having an effect on the body fat that you are going to be storing? So if you live in a cold climate, you may need more body fat than if you live in a hot climate, for example. What's the diet like that you're getting? What's the quantity, the quality? Do you have the right vitamins to make sure that your immune system is up to scratch? All these different things. What's your lifestyle like? Are you doing exercise? Are you smoking? Do you consume alcohol? Are you vaping? Are you doing things that are going to affect your phenotype, affect, for example, your body mass? So they could go down the route of that, or they could, because they love a bit of plants, they could look more at plants. And are you noting that if we're looking at a plant and we see, for example, yellowing of leaves, what is causing the yellowing of leaves? Is that low light levels? If we've got low light levels, we know those light levels are going to influence, they're going to be detected and then influence the gene expression. Is it that the genes that are needed for the chlorophyll production are being switched off? Because if there is low light levels, i.e. in winter, if there's low light levels, we're not going to want to have lots of chlorophyll production. We're going to see those accessory pigments because we're going to stop producing chlorophyll, because to produce chlorophyll, to maintain the chlorophyll, that is energy demanding and there's not that much light available. Is it that the availability of the mineral ions in the soil are having an effect? So if there's no magnesium in your soil, is that going to affect obviously the chlorophyll production? If there's no iron, is that going to affect the cofactor for the enzymes needed for the production of a pigment? Or is it a viral infection? Is there for example, the TMV? Have you got a problem in that you have the tobacco mosaic virus? Is it that there is a virus that has inserted its genetic information into the cells? It's hijacked the transcription translation processes. It's hijacked the machinery inside the cell, the RER. It's hijacked the Golgi. Whatever it is that's going on, is that therefore affecting the production? So your phenotypic variation that we're seeing is going to encounter or going to be influenced by the environment. So these are the things you have to know when it comes to inheritance. So are you confident with your monohybrid crosses? Are you confident with your dihybrid crosses? Are you confident with sex linkage? Are you confident with autosomal linkage? Do you know how to use a chi-squared a statistical test? Do you know about the definition of how these alleles are going to, these different forms are going to result in a gene pool within a population? So if we have a gene pool, and that gene pool is all of the alleles within the population, if we're looking at how those genes are going to flow, we're talking about the movement of genes from one population to another. So we've talked about how we can inherit the genes, but do you understand how those genes can then flow from one population to another? So if you have a island, so let me just do this again. If we have an island and that island is connected to another island, let's say Ice Age, there was a ice bridge where there was a habitat, there was a connection between two islands, and then as we have increased our global warming, we have resulted in an increase in our temperatures. Our sea levels have risen because the ice melted. We have now separated them. So if they have been separated, there is going to be no gene flow, okay? However, if we are flying somewhere, if we are using boats, if we are having some form of transport that allows for the migration of these organisms, that could result in the movement of these genes from one population to another. So that could allow for genes to flow. So if you are looking at anything to do with your alleles and the allele frequency and the gene pool, you could talk about natural selection, but they may actually be looking for your understanding of genetic drift. So if we have a population, size and it is significantly small, then there could be random mutations that happen, and those random mutations will affect the alleles that are going to be passed on. So patterns of inheritance, how these alleles are passed on, those alleles are passed on, but if we are talking about a small population, we're going to have a potential loss or favor of alleles, but this is completely by chance. This is completely to do with a natural thing rather, I shouldn't say natural, it's by chance rather than natural selection where they're being selected for. So genetic drift, when we talk about genetic drift, we either can talk about our bottlenecking effect. That bottlenecking effect is where by chance there is some thing that's happened, whatever that environmental event may be, that is going to cause this population numbers to drop dramatically. So if the population numbers are going to drop dramatically, that is going to reduce your genetic diversity. So that's module 4 and module 6 coming together. If we have a dramatic fall in our numbers, whatever that catastrophic event may be, it could be a flooding event, it could be a volcanic eruption, it could be a disease, unfortunately, that they're susceptible to. We have a decrease in the population. Whatever that decrease in the population is, whatever caused that fall, we don't know. Look at the stem of the question. But what's going to happen is many of those alleles are going to be lost. Our gene pool has been decreased. And so therefore, those survivors are the only ones that are going to be able to reproduce. And therefore, because they're going to be able to reproduce, the alleles of the survivors are going to be passed on. Now, remember that gene pool is very small. So because that gene pool is very small, therefore, the survivors are going to be the ones that reproduce. That could result in an increase in homozygosity because there's a lot of inbreeding. Those survivors could be related, and if those survivors are related, those alleles are going to be similar. How long, Imogen, is this live going to go on for? I'll go on for, what time are we at right now? 20 past. Wow, I've almost been going for 2 hours. I'll probably go till 11:30 because I've been going for 2 hours. And then I will be going live again tomorrow. But I need to break because I have one of my students coming to me later, and I don't want to lose my voice before the actual exam. So yeah, I'll probably do 10 more minutes and then I will stop after those 10 minutes. But it is on my YouTube channel if you want to go and watch it. So please do go and watch it from the start if you are looking for some revision before Friday. The founder effect, the other one which is linked to genetic drift. So genetic drift is a significant small population size, random mutations affecting which alleles are passed on. Small number of individuals start a new population. So my example above, where we had originally the bridge and then the bridge is going to disappear. If that is the case, then that could be an example of a founder effect, that we have a small population, a founding population. It's a new population and only a small number of the total alleles that were present. So for example, there was a question in 2019's AS paper, which was about the European wildcats and was about the Scottish wildcats, and it was talking about this specifically, and it was looking at you understanding how the genetic biodiversity can be affected and can be reduced if the population size decreases. So if we have a new population which has a small number, let's say they've got, 100 individuals. If I use that example, if there is 100 or even as little as 35 individuals, if there is a small population, how many of those population can actually reproduce? is there a large number of those 100 that are already past reproductive age? Are they immature individuals? So have they not reached maturation for reproduction? So there's lots of other things we have to consider when we look at the passing on of alleles. And that again links as well to if they were looking at sustainability and you're fishing, looking at the fact of if I have small fish that can escape out my nets, if I am going to do my fishing to feed the growing population, if I've got the small ones to be able to leave, the small ones are going to be able to get to that reproductive age. If they get to reproductive age, they can reproduce and that can then pass on those alleles. But what if the population, the small population that are actually surviving, only have the same type of alleles? That could result in an increase in homozygosity and therefore more inbreeding, could result in more problems. So these are sort of things that they could look at. Okay, so the other thing to know, if we're looking at, instead of genetic drift, if we're looking at natural selection, we have our key words that we should know for natural selection. So if I'm ever asked about, hold on, let me input here. If I'm ever asked about natural selection, so if I am ever asked about natural selection, oh my gosh, let's try that again. Natural selection, I would automatically bring in, excuse me, my keywords. So my keywords are mutation, variation, selective pressure, survival, and reproduction. Alleles passed on. Anyone who hasn't seen Grease 2 doesn't know that song. Mutation, variation, selective pressure, survival and reproduction. Alleles passed on, and many generations. And then if I'm looking at natural selection and I then, oh no, go away. If I'm then looking at the accumulation of these changes, that would therefore link to evolution being the accumulation of changes in allele frequency. Okay, so if I note that with my keywords, I should then look at the fact that if I have got natural selection, I should know that there will be some selective pressures, and those selective pressures could result in the changes within those allele frequency. So if there is a directional selection, that means that one of the extremes in the range of the variation is selected for and another is selected against. So that can result in a shift in the phenotype to what's known as a new optimum. And that could be the environmental change. So if I were talking about my peppered moths, I could talk about the Industrial Revolution, the soot and the particulates that went up into the atmosphere that coated the trees, and now the dark peppered moth is selected for and the light peppered moth is selected against. So therefore we have selection against a phenotype and selection for a phenotype, and that would be that we would find more of the allele frequency towards the melanistic peppered moth rather than the light one. Now, we've cleaned up our air, we've cleaned up our atmosphere, we're much better, we put less particulates out. So therefore, there is now a directional shift, which is showing this shift in the phenotypes to a new optimum when the environment changes. So that is directional. If we would see a shift, now that could be a directional shift that there is a phenotype which is selected for and a phenotype which is selected against. So we're moving to one of the extremes. If it is stabilizing selection, so if we're seeing some form of stabilizing selection, we would therefore see a favor of the intermediate phenotypes. Now, because we're talking about continuous variation, because we're talking about there being many intermediates, the extremes would be selected against and the intermediate would be selected for. So I've spoke about the birth weights on that one. That could be an example of selection. If you're looking at, for example, directional selection, I could imagine them to ask a question that looks at inheritance of, potentially, sickle cell hemoglobin. So if I have my, oh no, come away, I want this and I want this. If I have hemoglobin, and you can have normal hemoglobin, you can have sickle cell hemoglobin, or you can have, obviously, heterozygous individual, which is normal, has a normal and has sickle cell. So this person would be a carrier. This person would be unaffected. Oh, crumbs. If any of you have an iPad and have a hint as to how I can make it register my writing so it's not atrocious, because I know my writing is bad as it is, but this seems to make it look even worse. If we've got unaffected, I need to practice. That's what I need to do. Ah, see, it's just writing it. It's not registering. It's registering my wrist rather than the actual unaffected. Oh my gosh, right, whatever. Let's just put unaffected and sickle. So this one is sickle. So there we have our sickle, we have our unaffected, and we have our carrier. Now, what they could do is talk about, for example, if there is a problem with, and they're linking it to malaria. If there is a problem with malaria, malaria we know is caused by the protoctista. So we know it's caused by the protoctista. We know that the protoctista pathogen is carried in the vector which is the mosquito. We know that that vector will then feed on the human, and when it feeds on the human, it injects the pathogen into the bloodstream. And when it injects the pathogen into the bloodstream, it will then allow for the protoctista pathogen to be able to survive and thrive. Now, if you have sickle cell anemia, actually, you are protected against malaria. So it is better to have sickle cell anemia in places where malaria is an issue. And so if that's the case, then there has been a directional shift in places where we have malaria as being a problem, that there is more people that have sickle cell alleles, and therefore they're more likely to survive. If they're more likely to survive, they're more likely to pass on those alleles, and therefore their offspring are also more likely to survive. And so because of that, what that then means is we see an increase in the frequency of these alleles within that population. Now, with migration, what we can see is with migration, we therefore will then see those alleles, the gene flow of those alleles will go to populations where actually malaria is not a problem. And so if that's the case, then what we could see is an increase in those alleles being in a population where there isn't a problem with malaria. And so it could be a question along those lines. So malaria obviously is your module 4, communicable diseases, topic area you should know about. Hemoglobin is module 3, which could be in paper 3. However, sickle cell anemia is linked to inheritance. So it could be module 6 and module 4 connected by looking at the inheritance of the sickle cell allele and looking at how that could help in places where malaria is prevalent. So it really just depends where they're going to go. Right, guys, I'm going to stop there for today because I need to rest my voice and I've got some of my private clients. I hope that's been helpful and that's been useful. I am going to do a live again tomorrow. I'll probably go live at 9:30 again tomorrow. I'll do another 2 hours tomorrow, and I'll be looking at the other parts. I want to look at biotechnology. I want to look at manipulating genomes. I want to look at the nitrogen cycle as well. So I will go live again tomorrow. I'll do another 2 hours tomorrow for you all. Thank you to those of you who've been with me today. Thank you for those of you who have liked and shared and are watching my videos, because I do really appreciate it. Obviously these ones are my free lives. These are not the ones that I charge for. My group sessions are separate. So if you are a current year 12 and you're interested in group sessions, then obviously please do get in contact, because I am about to start the A2 for current year 12s moving forward. I'm starting that on the 22nd of June. Once my current cohort have left, then I will be starting the new set for my current year 12s who are moving through to A2, and for anyone else who'd like to join their small group. So that is available from the 22nd of June. And for those of you who are year 13 and you have your exam on Friday, I know it's hard. This is probably one of the hardest papers for most students because the content on genetics is difficult. Keep going with it though. Like me, I have this new setup. I'm trying, because I keep crashing my laptop, I had to buy a new one. I'm trying out a Mac right now. I'm using it with an iPad. My writing isn't great at the best of times, but I'm not used to using the iPad. I'm used to using my Hawaiian, and now I'm trying something new. The more I do it, the more practice I get, the more I'm okay with making mistakes, the better I will become. So keep going. You still have time to do the revision. Keep going, and hopefully you will be able to put some of this into practice in your paper on Friday. But good luck, guys, and I will see you all. If you join me again tomorrow, I'll see you tomorrow. If not, I will go live again on Thursday, the night before the exam on Friday. So guys, have a good one, and I will say goodbye.